Today, we turn our attention to the subject of stem cells, which has a lot of tangents to the topics that we covered in the first 3 modules, but it is also of such importance to the topic of tissue engineering that it merits some in-depth attention in a week-long module. And even then, we will only be laying the foundation for independent learning through research for those of you who are interested in this science. Stem cells are thought to be the holy grail of medicine. They are hoped to possess the power to alleviate all manners of suffering, whether it is caused by disease, injury, or genetic mutations. The age-old dream of regeneration is being animated by stem cell research. Scientists and bioengineers are recreating components of the body's various systems from raw materials that exist within. In tissue engineering or regenerative medicine, and both are interchangeable terms with subtle differences in connotation, the classical paradigm is to isolate and expand the donor's stem cells, and then to coax these cells to differentiate along a specific cell lineage to create a tissue replacement for eventual implantation in the recipient to cure a disease or replace a missing tissue. Stem cells have the potential to provide new and more effective treatments for diseases such as diabetes, heart disease, genetic diseases, neurological disorders like Parkinson's and Alzheimer's, and even cancer. They have the potential to repair debilitating injuries such as spinal cord damage. They have the potential to restore lost function, such as our senses of sight, and perhaps someday even to restore and regenerate lost limbs. Already they have enabled blind mice to see, paralyzed rats to walk, and monkeys suffering from severe Parkinson's disease to show dramatic improvements in their symptoms. We will see plenty of examples of this over the next 3 lectures. But there are also other uses of stem cells. For example, to create in vitro models of human disease that can be used in biomedical research to uncover and unravel the origins of this disease, or to test drugs and biological approaches to cure it. And we will also see some examples of this as well. Our objective over the next 3 lectures is to recognize the defining properties of stem cells, to identify the major differences between the different cell types, including embryonic and adult stem cells, to learn how researchers can isolate and characterize stem cells, to understand how stem cells can differentiate in a more specialized cell type, and to appreciate the potential clinical applications of stem cells in regenerative medicine and tissue engineering. So let's start with some definitions. What is a stem cell? A stem cell is an undifferentiated cell of a multicellular organism that is capable of giving rise to indefinitely more cells from the same type and from which certain other kinds of cells could arise by differentiation. A simpler definition is that it is a cell that has not yet acquired a special function. And implicit in these definitions is that stem cells can replicate themselves, or they can differentiate into many cell types. So what are the characteristics of stem cells? Recall that a stem cell exists in various tissue types in a microenvironment called as a niche. This is a concept that we will be discussing in just a few minutes. In the niche, stem cells are said to be quiescent. And one of their first defining properties is self-renewal. Self-renewal is a property in which a stem cell can divide to produce 1 or 2 daughter cells like themselves. Here's an example in which a stem cell produces 2 cells, one identical to the mother stem cell and the other a slightly differentiated cell, programmed to commit eventually down a specific lineage of a specific tissue type. This represents asymmetric cell division. So, what is the nature of the differences between the two daughter cells? Interestingly, these cells are genetically identical, but they read the genes in different ways. This has to do with a lot of factors related to different programs of gene silencing. Most importantly, perhaps, is the epigenetic regulation, which we will also be looking at towards the end of this lecture. So, if the daughter cell is genetically programmed to divide and produce identical dividing cells, these cells are said to be in transit amplification. And here's an example of that process in which one of the daughter cells undergoes transit amplifications to produce a number of clonogenic progenitor cells or precursor cells. So what you notice here is that a single stem cell, through one or a finite number of cell divisions, can generate many stem cells or progenitor cells that amplify to eventually differentiate and renew tissue. The pathway to differentiation usually involves the daughter becoming a precursor cell, which proliferates before it differentiates. Because the proliferation amplifies the number of differentiated cells eventually produced, the precursor cells are called transit amplifying cells. And as these transit amplifying cells undergo proliferation and more specification or differentiation, they lose their stemness and they acquire more of a differentiated phenotype. Notice here that the terms stem cells, progenitor cells, and precursor cells are often used interchangeably, but there are subtle connotational differences. Progenitor cells typically refer to a cell with greater developmental potential than a precursor cell, and stem cells are yet equipped with an even greater developmental potential than progenitor cells. So the concept of self-renewal is important. It implies that stem cells are mostly quiescent in the niche, and as a result of the process of growth and division doesn't deplete the pool of stem cells in the body or in a particular tissue. Therefore, most of our body's tissues harbor stem cells that contribute to tissue renewal, because most of the differentiated cells in these tissues have a finite lifetime, which could be measured in years, months, weeks, or even days and sometimes hours. For example, in the case of the brain, things turn over very slowly, while in bone the tissue turns over about 10% every year, and in blood the turnover rate is a lot faster than that. So all of our tissues are maintained by their stem cell compartments, and these compartments offer sources of stem cells that we can obtain and use for biomedical applications. Stem cells give rise to various cells when they acquire specialized function through the process called cellular differentiation. Stem cells are found in fetuses, embryos, and in numerous adult tissues. In a developing embryo, stem cells may differentiate into all of the specialized embryonic tissues. They originate from the inner cell mass of the blastocyst. In adult organisms, stem cells act as a repair system, replacing tissues damaged by disease or injury. They also maintain the normal turnover of regenerative organs such as blood, skin, and the intestinal tissues composed of epithelial linings, for example. Not all cells are created equal. Different stem cells possess different powers. Embryonic stem cells, for instance, have the capacity to recreate and repair any of the body's tissues and organs. Scientists often call these cells the gold standard of stem cell research, the mother of all stem cells. Adult stem cells, like those present in bone marrow, have the potential to repair some tissues and organs, but not all tissues in the body. This gives rise to the concept of cell potency. a general term which describes the cell's ability to differentiate into other cell types. The more cell types a cell can differentiate into, the greater its potency. Potency is also described as the gene activation potential within a cell, which, like a continuum, begins with totipotency to designate a cell with the most differentiation potential, pluripotency, multipotency, oligopotency, and finally unipotency. The term potency is taken from a Latin origin, potens, which means having power. Totipotent cells have the ability to divide and produce all of the differentiated cells in an organism. A good example of totipotent cells are zygotes, or even spores. In the spectrum of cell potency, totipotency represents a cell with the greatest differentiation potential, and the term toti comes from a Latin origin, totus, which means entirely. Next in the spectrum is pluripotency. Pluri is derived from a Latin source, plurimus, and it refers to a stem cell that has the potential to differentiate into any of three germ layers, endoderm, mesoderm, or ectoderm. Again, cell pluripotency is also a continuum. It is exemplified by the completely pluripotent cells that can be derived from the inner cell mass of the embryo. These are known as embryonic stem cells. We will also later be able to see that adult somatic cells, like a skin cell, can in fact be induced to become a pluripotent embryonic stem cell-like cell. Next, multipotency describes progenitor cells which have the gene activation potential to differentiate into multiple but limited cell types. For example, a multipotent blood stem cell is a hematopoietic stem cell, and this cell type can differentiate itself into several types of blood cells, like lymphocytes, monocytes, neutrophils, etc., but cannot differentiate into brain cells, bone cells, or other non-blood cell types. Oligopotency is a special kind of multipotency, and it is the ability of progenitor cells to differentiate into only a few cell types in a specific lineage. It is a degree of potency that is less than multipotency. Examples of oligopotent stem cells are lymphoid or myeloid stem cells. A lymphoid cell can specifically give rise to various blood cells such as B and T cells, but it cannot give rise to a myeloid progeny such as red blood cells. Finally, in the spectrum is a unipotent stem cell. This concept is a little vague, and it specifies that one stem cell has the capacity to differentiate into only one cell type. A good example, or perhaps a very close synonym for unipotent cell is a precursor cell. So for cells to renew themselves, they need to divide. And they can do so by a couple of different mechanisms that dictate the nature of the developing cells. They can divide asymmetrically to produce two daughter cells with different cellular fates. This is in contrast to normal symmetric cell division, which gives rise to daughter cells of the equivalent fate. Notably, stem cells divide asymmetrically to give rise to two distinct daughter cells, one copy of the original stem cell, as well as a second daughter programmed to differentiate into a non-stem cell fate. In principle, there are two mechanisms by which distinct properties may be conferred on the daughters of a dividing cell. In the first mechanism, the prospective daughter cells are inherently different at the time of division of the mother cell. Because this mechanism does not depend on interactions of cells with each other or with their environment, it must rely on intrinsic asymmetry. Intrinsic asymmetry divisions rely on the following mechanism. At mitosis, certain proteins RNA transcripts, and other macromolecules are localized asymmetrically to one half of the dividing cell. A cell can accomplish this through a variety of processes, such as localized molecule tethering, as well as molecular transport. Following this, the cell performs cytokinesis and divides into two. Thus, the asymmetrically localized proteins, RNA transcripts, and other molecules are inherited differentially to only one of the daughter cells, causing that cell to assume a separate fate from its sibling. Because these molecules ultimately determine the identity of the daughter cell, they are called cell fate determinants. In the second mechanism, the daughter cells are initially equivalent, but a difference is induced by signaling between the cells from surrounding cells, from a precursor cell, or from the surrounding microenvironment known as the niche. This mechanism is known as extrinsic asymmetric cell division. So an important factor in determining the self-renewal capacity of stem cells is a microenvironment known as the stem cell niche. It also somewhat dictates the fate of the dividing stem cell, and that contributes to the rejuvenation and regeneration potential of tissues. The stem cell niche refers to the microenvironment where stem cells can exist, which interacts with these stem cells to regulate cell fate. The word niche can be in reference to the in vivo or an in vitro system that represents a stem cell microenvironment. During embryonic development, various niche factors act on embryonic stem cells to alter gene expression and to induce their proliferation or differentiation for the development of the fetus and the different tissues within it. Within the adult human body, stem cell niches maintain adult stem cells in a quiescent state, but after tissue injuries, the surrounding microenvironment actively signals the stem cells to either promote self-renewal or differentiation to form new tissues. The niche is a complex interplay of short and long-range signals between stem cells and surrounding cells, their differentiating daughters, and neighboring cells. It is composed of secreted factor that could be endocrine, paracrine, or cell ligands. The niche also is composed of cellular interactions between the stem cells and other support cells like stromal cells, endothelial cells, and blood vessels, as well as progenitor cells that exist within the niche. There's also interactions between the stem cells and their extracellular matrix within the niche, and there's even some association with blood vessels that might even create a gradient in oxygen concentration that could be very important for the maintenance of a stem cell in a quiescent state, or triggering its differentiation into a more specialized cell type. One of the best characterized niches of stem cells is that of the hematopoietic stem cells, which exist in bone marrow. There is no conclusive evidence as to the precise anatomical identity of the hematopoietic stem cell niche. In one view, bone surfaces and the cells associated with them, known as osteoblasts, represent the niche. In another, the niche is localized to the walls of bone marrow blood vessels, or sinusoids. In reality, the structure of the bone marrow is so complex that vascular walls exist in very close proximity to the bone surface, and may in turn be lined at least on one aspect by bone cells. In addition, cells of osteogenic lineage exist in the bone marrow stroma. These are known as stromal cells, away from bone surfaces. So retention of a stem cell within a niche may determine the quiescence or self-renewal potential of hematopoietic stem cells, while egress from the niche may determine the exposure of hematopoietic stem cells to changing environmental clues, dictating their proliferation or differentiation into a hematopoietic cell progeny rather than a quiescent stem cell. Stem cells in the niche interact with surrounding cells as well as with the extracellular matrix, and these interactions that take place through transmembrane receptor proteins typically result in signaling that dictate a change in gene expression and potentially a transformation in the fate of the cell. One of the most important signaling pathways in hematopoietic stem cell compartment in the bone marrow is the Notch signaling pathway, which is essential for hematopoietic stem cell formation during embryogenesis and also required for hematopoietic stem cell maintenance. It is mediated by RBPJ kappa transcription factor activation, also known as CSL. The Notch receptor is a single-pass transmembrane receptor protein. It is a heterooligomer composed of a large extracellular portion, which is associated with another component, also composed of a short piece of extracellular protein, a transmembrane pass, and a smaller intracellular region, known as the Notch intracellular domain. Ligand proteins bind to the extracellular domain and induce a proteolytic cleavage by an enzyme called gamma secretase to release the intracellular domain of Notch, which then enters the cell nucleus to modify gene expression. The transcriptional factor target of Notch signaling is a transcriptional repressor known as RBPJ kappa. Absent Notch signaling, this transcription factor is basically repressing transcription of genes. And when Notch signaling is initiated, the intracellular Notch domain complexes with RBPJ kappa or CSL and another co-transcription factor called MAML1, and this results in activation of gene expression that are specific to the Notch signaling pathway, known as the HES and the HEY genes. Indeed, Notch signaling is a key factor in inhibiting differentiation of hematopoietic stem cells in the bone marrow niche. In studies done using genetic mouse models, it's been demonstrated that Notch signaling is active in hematopoietic stem cells in vivo and downregulated as these hematopoietic stem cells differentiate. Genetic models also allow us to introduce concepts such as inhibition or loss of function and gain of function. Inhibition or loss of function can also be introduced to cells using chemical compounds that could either inhibit a function of a specific protein down the signaling pathway or introduce a dominant negative form of a transcription factor. These are important tools in tissue engineering. Here are a couple of examples in which you can develop loss of function models by introducing, for example, a gamma secretase inhibitor to prevent the cleavage of intracellular Notch domain and eventually preempt the signaling pathway altogether. One can also interfere in this system by introducing dominant negative forms of a transcription factor CSL or RBPJ kappa and a dominant negative form of MAML1, the co-transcriptional activator. And introduction of any of these inhibitors or dominant negative proteins will result in abolishing the Notch signaling. And this would have profound effects on the cells depending on the nature of their differentiation stage. For example, the effect in mature cells is very different than that in hematopoietic stem cells, even though the signaling pathway is identical. And through methods like these, researchers have demonstrated that inhibition of Notch signaling leads to accelerated differentiation of hematopoietic stem cells in vitro and depletion of the hematopoietic stem cells in the in vivo niche compartment. Also, it's been demonstrated that intact Notch signaling is required for Wnt and beta-catenin mediated maintenance of the undifferentiated hematopoietic stem cells, but not for survival or re-entry into the cell cycle. And collectively, the data suggests that Notch signaling has a dominant function in inhibiting differentiation and providing a model for how hematopoietic stem cells may integrate multiple signals to maintain a stem cell state. In addition to cell-cell interactions, of which Notch signaling is only one example, there's also interactions with extracellular matrix components. The extracellular matrix, or ECM, is an intricate network of proteins that surround the cells and has a central role in establishing an environment that is conducive to tissue-specific cell functions. In the case of stem cells, this environment is the stem cell niche, and ECM signals participate in cell fate decisions. Cells express receptors that are members of the integrin superfamily. These are heterodimers, and in the hematopoietic stem cell compartment, hematopoietic stem cells express VLA4 or integrin alpha 4 beta 1 and VLA5 or integrin alpha 5 beta 1. These bind to fibronectin and osteopontin in the bone marrow ECM. It's been shown that binding to fibronectin regulates the expression of specific transcription factors that have roles in stem cell maintenance. Osteopontin is an important matrix glycoprotein that is exclusively produced by osteoblasts on the endosteal bone surface. It is a key adhesion component of the osteoblastic niche. OPN binds to VLA4, VLA5, and CD44 that are expressed by hematopoietic stem cells. This supports the adhesion of hematopoietic stem cells to the osteoblastic niche. In addition, it's been shown that OPN, or osteopontin, acts as a negative regulator of hematopoietic stem cell proliferation. It's been shown that it increases the cycling of hematopoietic stem cells when it is eliminated from the bone tissue in osteopontin knockout mice. The proliferation of hematopoietic stem cells was also suppressed by exogenous addition of osteopontin in vitro. Interestingly, the effects of osteopontin are not independent of other factors. For example, one of the factors that are known to stimulate hematopoietic stem cell proliferation is a hormone known as parathyroid hormone. And when the bone marrow niche in osteopontin knockout mice is stimulated using PTH or parathyroid hormone, there's an abnormal increase in the number of hematopoietic stem cells that is observed. And the thought here is that osteoblastic niche is activated. This leads to hematopoietic stem cell proliferation. And osteopontin in this context may function to restrict excessive stem cell expansion, which could eventually lead to depletion of the pool. So it is a very important ECM component of the niche. And the niche contributions to stem cell fate decisions is not limited to cell interactions or ECM interactions. There's a variety of cytokines and factors that are expressed. The association with the vascular cells is also important. And these are important concepts that we cannot cover in full in these lectures, but should be appreciated. So when a cell begins to differentiate in its niche, its differentiation often involves gene silencing. Recall that when we were discussing embryonic development, we had mentioned that the genetic material a cell contains is identical to the genetic material that it starts out with during embryonic development. We do not lose our genes, but many of our genes are silenced as the cells become more and more specialized. This is typically done through a process known as epigenetic regulation. Epigenetics is defined as the stable alteration in gene expression and potential that arises during development through the process known as differentiation and cell proliferation without altering the DNA sequence. It is a heritable change through modifications not affecting the DNA sequence. And there's been multiple evidence suggesting that the maintenance of lineage commitment of stem cells are controlled by epigenetic mechanisms, including DNA methylation, histone modifications which include acetylation and deacetylation, as well as regulation of ATP-dependent remodeling of chromatin structures. Now recall that chromatin is the combination of complex of DNA and proteins that make up the contents of the nucleus of the cell. The primary functions of chromatin are to package DNA into smaller volume to fit into the nucleus, to strengthen the DNA to allow mitosis, and to prevent DNA damage, but more importantly also to contribute to controlling gene expression and DNA replication. The primary protein components of chromatin are histones that compact the DNA. And when a cell begins to undergo differentiation, the transcriptional active chromatin regions are converted to inaccessible domains. In a stem cell, transcriptionally active chromatin regions tend to be hyperacetylated and hypomethylated. If the cell begins to differentiate and the region of DNA of this gene needs to be silenced, chromatin remodeling enzymes, including histone deacetylases and ATP-dependent chromatin remodelers, begin the gene silencing process. One or more of these activities may recruit DNA methyltransferase, an enzyme that results in DNA methylation, followed finally by recruitment of methyl CpG binding protein. This results in compaction of the chromatin structure of the DNA, effectively making the region of the DNA inaccessible for transcription factors to bind to the gene and affect gene expression. And this eventually leads to gene silencing, which is a very important hallmark of differentiation of cells, in addition to gene expression, obviously. The interesting thing is that as embryonic stem cells undergo differentiations, the level of DNA methylation increases. And this is in agreement with the discussion we just had in the previous slide. If you look, for example, at the methylation state of primordial germ cells such as sperm and egg, they start out with, during development, being hypomethylated. And as they specialize as a sperm and an egg, they then undergo de novo methylation in a developing germ cell. The fertilized cell itself begins to undergo a quick demethylation in the early embryonic stages until it forms a blastocyst, where the cells are extremely hypomethylated and the expression of many genes are enabled. As the cells begin to differentiate into somatic cells during embryogenic development, they undergo de novo methylation in somatic cell lineages. And this methylation or methylated states continue to be maintained throughout adult tissue life. Some of the support structure in the embryo, like the placenta and the yolk sac, also undergo some methylation, but that is much less compared to the tissues that actually contribute to the formation of the embryo proper. So I'm going to stop here today, and this is a nice segue that will take us into embryonic stem cells, which will be the opening topic of next lecture. If you have any questions, please try to read the supplemental material that I provided, and feel free to email me. We'll have the chance to go over some of your questions on Monday when I get back. Thank you.